Hello, everyone, and welcome to today's webinar, In Situ Single-Cell Transcriptomic Imaging in Formalin-Fixed Paraffin-Embedded FFPE Tissues with MERSCOPE. I am Marie Stone of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Vizgen. To learn more, visit vizgen.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer any questions as we have time for at the end of the presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speaker, Jiang He, co-founder and director of scientific affairs, Vizgen. Jiang, you may now begin your presentation.
Thank you so much for the introduction. And hello, everyone. My name is Jiang. Today, I'm super excited to share our latest progress in single-cell spatial genomics and specifically in situ single-cell transcriptomic imaging in FFPE samples. So for today's presentation, I'm going to cover two topics. First, I will give an overview of our platform technology, and then I will explain our recent advancement in imaging FFPE samples with MERSCOPE. As many of us know, biological tissues are very complex and made of many different cell types that express genes quite differently. And over the past 2 or 3 decades, sequencing has transformed our understanding of biology and really enabled us to characterize biological systems at unprecedented scale and resolution. However, as you can imagine, sequencing has fundamental limitations. It is difficult to use bulk or single-cell sequencing to infer where individual cells are or how they interact with each other, and our understanding of biological systems thereby is far from complete. In recent years, a new field called spatial genomics emerged, which really aims to add the critical spatial component back to the picture. And with spatially resolved single-cell genomics, we are now witnessing another revolution in genomics research. And as a company, Vizgen is bringing the first true single-cell spatial transcriptomic platform called MERSCOPE to the market. MERSCOPE is built based on a foundation technology called MERFISH, which enables simultaneous imaging of hundreds to thousands of different genes in a single run with high sensitivity and accuracy. It can also scan very large imageable area up to 1 square centimeter with very fine spatial resolution and no downstream need of sequencing. So this is an all-in-one user-friendly solution, which includes the upstream sample prep related reagents, instruments, and downstream visualization software. Highlighted here on this slide is the instrument that performs data acquisition and data analysis. And in the next few slides, I'm going to walk you through the principles of MERFISH technology first. So MERFISH stands for multiplexed error-robust fluorescence in situ hybridization. It is built based upon a RNA imaging technology called single-molecule FISH. And as shown on this slide, to perform SM-FISH, RNA transcripts of interest are labeled by fluorescence detect oligoprobes. And by directly counting the number of RNA species that fluorescence, we can then really determine the expression profile for each individual gene, as you can see from the image on the right. So SM-FISH is very well known for its sensitivity and often considered as the gold standard for quantifying RNA copy numbers. The limitation for SM-FISH, however, is that the plexing level is very limited, often less than 5. And to overcome this big limitation, MERFISH implements a novel complementary barcoding scheme that really enables simultaneous imaging of hundreds to thousands of different RNA species. So I'm going to use this schematic here on the slide to illustrate how MERFISH works. To perform MERFISH, we will first assign a set of barcodes to different genes of interest. And using gene 1 on this slide to illustrate the principle here, we will first label the target RNA species with a type of oligoprobes that contains different barcodes. You can see that it has a targeting region that will be able to bind to different RNA species, and it also has the handout that has readout sequences. Then we're going to flow in a subset of readout probes that is fluorescence detect, and then it will bind to its corresponding readout sequences on the handout region. And if there is a binding event, then it will show up as a fluorescent on signal, and then it will decode as 1. And if there is no fluorescent signal, then it will decode as 0. And then we will extinguish the signal and then flow in another subset of fluorescently labeled readout probes again. And then again, you can see in the second round, if there is a binding event, there is a fluorescent signal, and then it will decode as 1. And then if there is no signal, then it will decode as 0. So essentially, by repeating this cycle, we're building a series of 1s and 0s. And by matching with the pre-designed barcode, then it really allows us to resolve the identity as well as the quantity of different RNA species. So this barcoding scheme is further designed with error robustness because we only use a small subset of barcode in the available barcode space. And barcode sequences actually also have enough space in between. So incorrectly assigning a barcode is very unlikely. So if there is a readout error, this system can actually detect and correct it to the nearest barcode. And that's what's making MERSCOPE so accurate and consistent. So with this complementary labeling, sequential roundup imaging, and error-robust barcoding, MERFISH really enables researchers to now directly visualize gene expression in situ. As you can see from this animation here, you can zoom in and zoom out to really see the gene expression from whole tissue to single cell and even subcellular resolution. So there are a couple of key advantages of MERSCOPE platform, and I'm going to sort of walk you through one by one. The first one is that it is a truly quantitative measurement across a very large dynamic range of gene expression profile. So on this slide, what you are seeing here is a comparison of MERFISH data acquired in the mouse brain tissue with bulk RNA sequencing. And each dot represents a gene. The y-axis, you're seeing the copy number per cell counted by MERFISH, and x-axis, you are seeing the FPKM values from bulk RNA sequencing. So you can see that in general, MERFISH has excellent correlation with RNA-seq, with correlation coefficient around 0.88, and this linear range is spanning more than 4 orders of magnitude. So this large range is very important to non-nodally expressed genes to be accurately profiled. If you look at this, not only we can profile genes at high FPKM value, but also lower FPKM value very accurately. So the second key feature for MERSCOPE is that it has exceptional high detection efficiency. So as I mentioned before, SMFISH is often considered as the gold standard for quantifying RNA copy numbers and has single molecule sensitivity. And as a technology built based on SMFISH, MERSCOPE maintains that high sensitivity of SMFISH. So on this slide, I'm showing you the comparison of performance between MERSCOPE and an array-based spatial genomics platform in matched brain samples. So this is measuring hundreds of genes in the mouse brain. And for the same genes measured, I'm showing a single one here called OPRD1. This is a GPCR. You can clearly see where this gene is distributed in the mouse brain. However, if you look at the image at the bottom with the array-based platform, you only see a couple grid spots. And as you zoom in into individual field of views with MERSCOPE, you can clearly see which individual cell expressing OPRD1. On the other hand, when we look at the image on at the bottom with the array-based platform, since the grid spot size is around 55 microns, and in between there's a lot of dead space, thereby you don't really have any true single-cell resolution. And when we compare these two datasets, we consistently found that MERSCOPE has detects 70 times more transcripts per gene versus the array-based platform. So just really think about the difference in terms of sensitivity here and how much information you will actually lost if you use a technology that doesn't really capture much of the RNA transcripts. The third key feature I wanted to highlight is that MERSCOPE really features true in situ detection at single cell resolution. So on our platform, MERSCOPE uses high NA60X objective to capture all the images with imaging resolution down to 100 nanometers. Thereby, it offers true single cell resolution. And by combining immunofluorescence to label the cell boundary, MERSCOPE enables true cell atlas. So in this cancer cell paper published last year in 2021, for example, the authors used MERFISH to characterize different cell types in glioblastoma. So as you can see from the heat map in the middle, so this is showing the gene expression profile in the identified cell types. You can see that MERFISH was able to really recapitulate the gene expression features in the identified cell types. It almost looks identical to the single-cell RNA-seq characterization though. But since MERFISH is an in situ measurement, it does offer information that single-cell RNA-seq cannot provide. And this is enables the researchers now to study the interaction of macrophages to subtypes of cancer cells. And specifically, they measure the mean number of macrophages in 30 nearest cells to the MES like glioblastoma cells. And specifically, they found enrichment of macrophages adjacent to this subtype of glioblastoma cells in situ. So you can imagine that such findings really demonstrate that the power of in situ single-cell transcriptomic profiling to uncover and really characterize cell-cell interaction, because without this, such a finding will almost be impossible purely based on the single-cell RNA-seq analysis. Finally, MERSCOPE features extremely high cell throughput, and as an imaging-based technology, the number of cells profiled is directly related to the tissue area imaged. It is very easy for MERSCOPE to scale up the cell throughput by targeting more regions of interest to image. And specifically on MERSCOPE, it supports profiling a square centimeter sized tissue with hundreds of thousands cells can be captured in a single run. So for example, on this slide, we use MERSCOPE to profile 483 genes across the mouse brain. And what you're seeing here on the right in the zoom in is a single field view. Each single field view is 200 by 200 microns. And we type hundreds to even thousands of images to capture the whole brain section. And what's really nice about this system is that no follow-up sequencing is needed. You get all the information from an imaging run. So the technology has been around for a number of years now. It was invented by our scientific co-founder, Dr. Xiaowei Zhuang at Harvard University back in 2015. And since the initial publication in the Journal of Science, it has been validated by a long list of publications now and brought many transformative discoveries. And across the globe, there are already many institutions using MERFISH, others through their own implementation or through Vizgen's commercial efforts. So MERFISH has been widely used in many disease areas now, and in the next two slides, I'm going to highlight two studies here. For example, Dr. Ben Hamper's lab at Washington University at St. Louis used MERSCOPE to profile a panel of 243 genes in mouse kidney. An adjacent kidney slice was profiled by an array-based platform. And on the slide, I'm highlighting one gene called SLC12A1 here. So this is the kidney-specific sodium-potassium chloride co-transporter. And hopefully by just looking at these two pictures on the slide, you can see the striking difference in terms of the detection efficiency and resolution. You can, with the array-based platform, you only see a couple grid spots, while with MERSCOPE, you can clearly see where SLC12A1 is expressed and the elaborate structure is labeling in the mouse kidney. So in other words, the high detection efficiency and resolution provided by MERSCOPE really enables spatial profiling of genes inaccessible by other spatial technologies that is available on the market at the moment. So in a separate study published in Science back in 2018, MERFISH was used to profile 155 genes for a total of almost 500,000 cells in the mouse hypothalamus region. And the authors were able to identify 75 cell clusters in this brain region. So this is shown in the t-SNP plot here. You can see all the major cell classes labeled in different colors. And in this paper, the authors also performed single-cell RNA-seq and benchmarked the performance of MERFISH versus single-cell RNA-seq. And again, you can see quantitatively, MERFISH agrees very well with single-cell RNA-seq. All the major cell types found by single-cell RNA-seq were recapitulated by MERFISH measurements, as you can see from this correlation plot here. So as the cell position information is recorded in this measurement, MERFISH further helped reveal the cell organization in mouse tissue as well. So showing here in the middle now, these colorful dots indicate different cell classes identified by MERFISH. And from anterior to posterior, I hope you can really see how different cells are distributed in this brain region now. And furthermore, by including genes that can report neuronal activity, and in this specific case, the authors included a gene called C4. So they were able to really selectively identify neuronal types that are activated during individual social behaviors, such as parenting behaviors or mating behaviors. For example, showing here, this bar represents the neuronal type that is selectively active during parenting behaviors. And with the spatial information recorded in this measurement, you can now map out where those individual cells are now in complex tissue. So this study really highlights the power of MERFISH to perform true cell atlasing and to map cell types, characterize cell function and state in the complex tissue. So within Vizgen, we are keep pushing the limits of the technology by expanding the application areas. We have proven MERFISH in more than 30 different mouse or tissue or human tissue types now, and compatible sample types demonstrated so far include cultured cells, fresh frozen, fixed frozen tissue blocks. And showing here on this slide, you can see a couple of examples of different tissues we imaged in mouse or human. However, there is a big question unanswered. Can MERSCOPE be used in FFPE samples, which is one of the most widely used clinical sample types? So in the second half of my presentation, I'm going to focus on discussing MERSCOPE imaging in FFPE samples. And to give some context of why this application is very important, I'm going to use this slide to illustrate the pros and cons of different tissue preservation methods. So as you can see here, so fresh frozen tissues could really preserve DNA or RNA better, and it could even be compatible with single-cell sequencing analysis. However, the tissue blocks usually need to be stored at -80, and this is a very costly solution and often not available for clinical practices. On the other hand, FFPE, or so-called formalin-fixed paraffin-embedded samples, so they are widely available, and it could preserve the tissue morphology better. And around the world, there are banks of archival FFPE samples. So if we can really unlock the use of these FFPE samples, it will really enable and in more and more new discoveries made in in biology. However, there are challenges of actually working and perform single-cell analysis with FFPE samples. For example, heavy cross-linking can make it very difficult to access the RNAs inside a FFPE sample. And separately, RNA tends to be more fragmented in FFPE samples. And these challenges really require a substantial change of workflow in order to perform single-cell spatial genomics measurement in FFPE samples. So to overcome these challenges, at Vizgen, we have developed a new proprietary chemistry to facilitate MERSCOPE imaging in FFPE samples. And this is based on our first generation frozen protocol. The major changes are sample prep workflow, and thereby it is compatible with existing MERSCOPE platform and our portfolio products. So to evaluate the performance of this technology, we have chosen FFPE mouse brain for MERSCOPE imaging because it has very well annotated in situ gene expression profile, and thereby we can more accurately evaluate the performance of our chemistry in detail. So showing here is 483 gene panel imaging in the FFPE mouse brain, and all the detected transcripts are shown across the tissue. And in the middle, you are also seeing a zoomed single field of view showing the distribution of all transcripts at single cell resolution. So overall, we were able to detect around 45 million transcripts in around 55,000 cells in this single measurement. And the mean transcript count per cell is around 816. So this is comparable to our fresh frozen data sets released publicly. So if you're interested, we actually had a data release program that had the mouse brain receptor map released, and this is something that you will be able to download from our website, and it's using the same gene panel to perform the benchmarking in the FFPE study here. So furthermore, the genes profiled by MERSCOPE showed expected spatial distribution in the FFPE mouse brain as well. So showing here at the bottom, I'm showing 4 genes GFAP, CNR1, DRD2, and ADGRA1 profiled by MERSCOPE or by any institute's in situ hybridization. So hopefully you can really see that for all 4 genes, it has the expected spatial distribution pattern very nicely captured by MERSCOPE. So this really speaks that the new chemistry is working very well. And next, to more systematically characterize the performance of MERSCOPE FFPE workflow, we started to perform a couple of benchmarking to really characterize its performance. So we benchmarked the acquired data in FFPE with bulk RNA-seq and MERFISH data acquired by fresh frozen workflow. So as shown by this correlation plot here with bulk RNA-seq, MERSCOPE FFPE workflow is highly quantitative and with correlation coefficient of 0.88. And you can see that it also maintains the linear correlation with a very large dynamic range of expression, more than 4 orders of magnitude, similar to the range that I discussed a few slides ago. So this is also highly reproducible measurements, and in biological replicates, we almost see perfect correlation here, as you can see from this correlation plot, or almost with correlation coefficient around 1. So furthermore, when we compare the count detected by FFPE workflow in FFPE samples versus those that are detected by fresh frozen protocol in fresh frozen samples, we also found excellent correlation with correlation coefficient around 0.95. So furthermore, MERSCOPE's high spatial resolution allows true cell atlas, and we are able to really perform the cell type identification. And using this, it really allows us to now characterize, can we recapitulate the cell types identified by fresh frozen workflow in FFPE samples or not. So I'm going to play a short video here. Showing here is the UMAP identified by this panel of 483 genes. And then this animation is going to show the spatial distribution of all the different cell types identified by FFPE workflow at the top, or the fresh frozen workflow. And as we are looking at different leading clusters, I hope you can really appreciate how FFPE workflow is able to recapitulate all the major cell types identified by our fresh frozen workflow. So in a short summary here, it looks like our FFPE workflow allows very large dynamic range of expression profile for profiled genes, more than 4 orders of magnitude. It is highly sensitive and accurate measurement and comparable performance to fresh frozen workflow as well. So having established the performance in the sort of our benchmarking sample mouse frame, next we wondered if MERSCOPE FFPE workflow can be used in other tissue types other than mouse brain or not. And since most FFPE samples are coming from human samples, we next tested the samples in FFPE human colon cancer with a panel of 244 genes. And in this single slice, we were able to detect 135 million copies. And again, as you can see from the picture here, I highlighted 7 genes here, ACTA2, which labels the smooth small mass-actin CD1, 63 macrophage marker, PCAM, the endothelial marker, keratin, mucin, and TGF-beta. You can clearly see the very beautiful structure that these genes are highlighted in the colon cancer. We also benchmarked performance versus bulk RNA sequencing. And as you can see here, each individual gene are indicated in this blue dot here, and the y-axis is the MERFISH counts, and x-axis are FPCAM values. And you can see the correlation coefficient of 0.81 here. So to demonstrate the wide applicability of MERSCOPE in the other samples, we decided to perform MERSCOPE imaging in more than 10 different samples. And showing here, so again, we will sort of really extracting the correlation coefficient with the correlation plot here, similar to this value I'm showing here, and just plot all the correlation coefficient with bulk RNA-seq in a bar chart for more than 10 different sample types, including breast cancer, colon cancer, kidney, pancreas, stomach, and such. And you can really see that across all the samples, it all has very excellent correlation. So the MERSCOPE FFPE workflow is compatible with proteins co-staining as well. So on this slide, we performed 6 protein imaging together with a panel of 347 gene panel measurements. So on the left, you're seeing 3 out of the 6 proteins here, pan-CK, SMA, E-cadherin. And on the right, you're seeing the distribution of all transcripts. And in this measurement, around 70 million counts were detected with a panel of 347 genes. And at the bottom, I'm zooming into one specific region. And on the left, again, you're seeing this very beautiful staining done by the proteins pan-CK, SMA, and E-cadherin. And on the right, you're seeing the overlay of all transcripts on top of the protein staining. So having established the performance of MERSCOPE in different samples, in the next few slides, I'm going to showcase a couple of studies that was performed in FFPE samples. And the first one is to really map the tumor microenvironment in FFPE human ovarian cancer. So this was done by using a panel of 500 genes. And in this single slide, I'm highlighting only 8 genes here. So in this measurement, we were able to detect 174 million RNA transcripts. And hopefully you can really see the beautiful spatial distribution of these 8 selected genes here. And to really illustrate what the data quality looks like, I'm going to zoom into the data and show you 2 animations here on this slide. So first, I'm going to zoom into this ovarian cancer and only highlighting these 8 genes here. Then I'm turning on 500 genes and further zoom in, and hopefully you can see the distribution of individual transcripts at single cell level. And then I'm zooming out now and then turn off these 500, only showing 8 RNA transcripts. And hopefully you can really see how the data looks like from across the tissue down to single cell resolution. So since we were able to perform cell boundary staining, which is antibody based, that allows us to segment the cells and perform single cell analysis. So on the right, we basically identified different cell types and projected them in a UMAP, and then also show the spatial distribution of identified cell types in space as well. So hopefully in this animation here, you can clearly see how different cell types, where different cell types are distributed. So it purely speaks the power of using MERSCOPE FFPE workflow to sort of perform cell atlasing in complex tissue like cancer. So next, MERSCOPE was used to image an archival FFPE human melanoma samples, and this was profiling a panel of 244 genes, and this sample was more than 5 years old. And on the top left, I'm only highlighting 6 selected genes out of these 244 gene measurements. For example, ACTA2 labeled as muscle-actin CD4 for the T cells, CD4 T cells, and CD79B for the B cells. Keratin labeled in the epidermis here, and hopefully you can really see the beautiful structure that these genes are highlighted here. And on the top right 2 images, this is showing the antibody staining for the cell boundary. Hopefully you can really see the very nicely defined cell boundary using our cell boundary kit. And with that, it allows us to really segment individual cells. So on this image here, you're seeing the polygon shapes drawn on top of the cells drawn by our default cell segmentation algorithm. So again, this allows us to now sort of identify different cell types and perform UMAP clustering. And you can see the different clusters here. And then we were able to project these different cell types in space. And I'm highlighting 3 different cell types here, for example, CD4 T cells, endothelial cells, and keratinocytes in the melanoma. So showing really, really unique distribution here. And in this measurement, actually 156 million RNA transcripts were detected. So the last example that I'm showing here is MERSCOPE imaging in FFPE human lung. And on the left, I'm showing the spatial distribution of 5 selected genes called ACTA2, AGER, and macrophage marker, CD163, mucin, and SPFT-A2. So we were also able to perform cell clustering as well and project the identified cells in space. So these different color dots now, what you're seeing is the identified cell types in the human genome. So overall, we were able to detect 70 million RNA transcripts in this human genome size. And in this zoomed-in image here, hopefully you can see, for example, these mucin-4 positive cells, namely the bronchia, surrounded by the ACTA2 positive fibroblasts. Then you can see the magenta, the macrophages, and then you see the type 1 and type 2 cells labeled by AGER and SFPA2 here as well. So overall, we have demonstrated MERSCOPE imaging in 15 FFPE sample types, and here are a few representative MERSCOPE images on different tissue types. For example, human lymph node, tonsil, non-cancer, skin, and kidney. And I hope really these images illustrate the spatial architecture of different tissue types and how important it is to have a really highly sensitive and versatile tool to profile gene expression and cell types. And around the globe, there has been researchers already adopting MERSCOPE FFPE workflow in the research. For example, the pharmaceutical company Compugen in Israel used MERSCOPE to study the immune tumor microenvironment in colon and ovarian cancer. And in Broad Institute, Neil Cohen's group is using this to study the colon cancer as well. And these are some of the posters that they have recently been presenting. So I also want to mention that Vizgen has a data release program, and we have released 2 data sets. So on this slide, this is a recent mouse liver data set measuring 347 genes, more than 1 billion transcripts. And we're actually going to have a FFPE showcase data release coming soon as well. So if you're interested, so please do sign up this data release program. And to recap what I have just shared today, so here is a quick summary of MERSCOPE advantages. First, MERSCOPE has high multiplexing power. We can measure hundreds to thousands of genes simultaneously in a single sample. And our default offering is custom gene panels, and it also is compatible with protein imaging as well. Secondly, MERSCOPE has exceptional spatial resolution. We can localize RNA with subcellular resolution, and this high resolution is preserved across the entire tissue. Third, MERSCOPE has highest sensitivity for detecting identify different RNA species, and it really enables us to detect lowly expressed but functionally important genes very easily. And furthermore, MERSCOPE has very high cell throughput. We can profile a square centimeter sized tissue with hundreds of thousands of cells measured in a single experiment with no downstream sequencing needed. So you basically get all the information from a single imaging run. And finally, MERSCOPE is very flexible, and we can perform MERSCOPE on almost all tissue or sample types. So in summary, I hope this presentation gives you a good overview of MERSCOPE platform and how we can actually perform FFPE imaging, MERSCOPE imaging in FFPE samples. And we're truly excited about the potential of our technology, and would love to hear how we could potentially help with your research as well. So with that, I wanted to thank you for your attention, and if you have any questions, please let me know or send any inquiries to the email address below. Thank you.
Thank you, Jiang, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what is the recommended RNA quality for a sample?
Yeah, so for fresh frozen or fixed frozen samples, we use RNA integrity number RIN score as a guideline for sample QC. And for frozen samples, RIN the best to be above 7, in between 5 to 7. It also will give good data, but it may have some compromised quality. So below 4, we don't usually recommend for frozen samples. On the other hand, for FFPE samples, we use a different metrics to evaluate RNA quality, which is called DV200 value. So far, we have mostly worked with samples above 40% DV200 value. So that will be so far our current guideline.
Thank you. Next question, do you need a different instrument?
So to work with FFPE samples, you don't need an additional instrument. So the major changes are the sample prep steps. So everything happens on bench, and you can use the same gene panel, the same instrument to work in FFPE to work with FFPE samples.
Thank you. Next question, how long is the target probe?
Yes, our target probe are around 30 nucleotides per probe. So yeah.
Okay. Next question, is it possible to use this technology with non-human and non-mice samples?
Yes, absolutely. So we actually already have early adopters using MERSCOPE on rats, on monkey samples, and there are even some groups trying plants. So as long as there's an annotated transcriptome available, we will be able to design probes against those RNA species and then perform MERFISH.
Thank you. Next question, what is the maximum area that can be measured per experiment? Does it always cover a full coronal section or only sub-area is preferred?
So yeah, on MERSCOPE, the total imageable area is up to 1 square centimeter. So this imageable area is actually larger than the coronal section of the mouse brain that showed here. I'd say to put that into context, that 1 square centimeter is probably 1.5 whole coronal section of the mouse brain. If it's right in the middle line of the sagittal brain region, it may be just covering around 1 square centimeter. So indeed, it will be able to cover the full coronal section and even provide, there is still some extra ROIs that you can select for other tissues if needed.
Thank you. Next question. Can you combine MERSCOPE with regular histology, such as visualizing innate GFP or antibody staining prior to MERFISH?
Yes, absolutely. So to image GFP signal, what we usually do is to use an antibody and against the GFP, and then use oligoconjugated secondary antibodies to recognize that. So it's possible, and you don't need to run it on an adjacent slide. You can do the antibody staining on the exactly same slides.
Great, thank you. Next question. Is the proprietary prep chemistry for FFPE destructive? That is, can the tissues be used for sequencing or other downstream techniques after MERSCOPE?
Yeah, so after the MERFISH is done, so in the sample, the final process sample, what's left behind is usually the DNA and RNA and oligos. Thereby, for downstream sequencing, you can actually take whatever is left in the sample and then do the downstream sequencing. So internally, we have not usually performed that kind of downstream sequencing, but we believe this is possible.
Thank you. Next question. FFPE has high autofluorescence. How does MERFISH overcome that?
Yeah, so we actually have developed our own method to remove autofluorescence. And this is not just related to FFPE, but also to the fresh frozen or fixed frozen sample types. For example, like across a variety of different human tissues, even in fresh frozen states, it will have some autofluorescence. And within our protocol, we do have two different approaches of removing the autofluorescence step. One is in our autofluorescence quenching step, and then the other one is our tissue clearance step.
Thank you. Next question. How many proteins maximum can be detected together with RNA?
Yeah, so for protein co-staining and imaging on MERSCOPE, this is actually a product that Vizgen as a company will be releasing in the coming months. So it's coming to the market very soon. So this is our oligoconjugated secondary antibodies, and it will be able to support a max up to 6 protein co-staining. So a researcher can choose whatever primary antibody coming from these different host species and then use the secondary antibody for protein co-detection.
Thank you. Next question. Where can we download the data sets?
So if you go to our website, there will be a data release program. So far, there are two data sets available. One is the mouse brain receptor data set, and the second one is the mouse liver data set. We will be posting our FFPE data sets, which covers quite a few different cancer types using a gene panel of 500 genes, and it has many. It's basically our immuno-oncology panel where you will see a lot of immune cell markers, cancer cell markers, and checkpoint inhibitor and their receptors and cytokines and such. They are in this gene panel. So if you go to our website and register for that, you will receive notifications for the next data release. And after this, we will actually continue to roll out more data coming from different tissue types. So if you work in a different research area also, I would highly recommend you to look for that resource as we continue to roll out more data.
All right. Next question. When will the MERSCOPE FFPE be available?
So the FFPE sample prep kits will be released later in the year, in a few months, I'd say around Q3 this year.
Okay, thank you. Next question. Do current panels designed for fresh tissue work with the FFPE samples?
Yes, so the gene panel used for frozen samples, the same gene panel can be used on FFPE samples as well. So you don't really need to redesign a gene panel.
Thank you. Next question. Have you tried MERSCOPE in FFPE human brain tissue?
Yes, so we have tried both fresh frozen and FFPE human brain, and both works very well.
Okay. Next question. Can you detect non-coding or non-poly A transcripts in FFPE?
So that is something that we have not very rigorously looked at yet. So for non-coding RNAs, MERFISH has been demonstrated to be able to detect. So our new chemistry will be able to detect sort of non-poly A tailed mRNA for sure. But we haven't specifically looked for the genes in that category though. But we believe our chemistry will be able to address and look for targets of that kind.
Thank you. Next question. With the neuroblastoma tissues, we see high fluorescence. How do you take care of the autofluorescence?
Mm-hmm. Yeah, I think this is a great question and really relates to the previous background question asked before. So indeed, in brain, lipofuscin and such is one of the major concerns and causes a lot of background. And the two steps that mentioned, the autofluorescence quenching and clearing, in general, can help remove all the backgrounds. And you can actually, if you're interested in seeing how this performs, you can go to our website and download our tech note. We do have a session on how to address the autofluorescence background.
Thank you. Next question. What is the maximal number of genes to be identified in one tissue sample with MERFISH theoretically?
So, so far, the highest number demonstrated through academic publication from our scientific co-founder, Dr. Xiaowei Zhuang's lab, is more than 10,000 genes in cells. And recently, her lab also published posted about a paper demonstrating human brain imaging with 4,000 genes in tissue. So that's the number that has been demonstrated so far in publication.
All right. Next question. I have heart tissue fixed with 4% PFA and was embedded in paraffin. Can I use these samples for MERSCOPE?
Yes, so the FFPE sample prep, the histology process is actually a very standard protocol. So we don't really have any harsh requirement of how the tissue should be fixed or how it should be embedded in paraffin. So this is compatible.
Thank you. Next question. When photo bleaching the FFPE sections, is there a risk of disintegrating the tissue section? Any measures that will need to be taken?
So for removing the autofluorescence using our autofluorescence quenching device, we actually, when we compare this in our frozen samples, we don't really see a major impact without using this in certain samples that doesn't have autofluorescence. So we don't believe it's disrupting the targets inside the tissue.
Thank you. Next question, what's the maximum thickness of FFPE sections for MERFISH?
So for now, we generally recommend basically what the immunohistochemistry field, IHC field, usually do, which is 4 micron thick or 5 micron thick. But if you wanted to go slightly thicker, it is also possible. So this is a little bit different from the frozen tissue. Frozen tissue, people generally cut around 10 micron thick. FFPE in the field usually is cutting 4 to 5 micron thick. So we are basically following the general guideline in the IHC field.
Thank you. And we will wrap up with that final question. Thank you again, Jiang, for your time today and your important research. We would also like to thank LabRoots and our sponsor, Vizgen, for underwriting today's educational webcast. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions we did not have time for today and those submitted during the on-demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.